So the second lecture on the topic of the muscular system is going to focus on muscle contraction and the energy systems that fuel muscle contraction. So we're going to take the structures that we learnt about in the previous lecture and look at the roles that they play in helping our muscles contract. So a few more learning objectives to cover in this lecture, although a few of these learning objectives will be quite short. We're going to define what a motor unit is. We're going to describe the events that occur at the neuromuscular junction in excitation, contraction coupling and in cross-bridge cycling, which together all make up muscle contraction. We'll look at the factors that influence force of muscle contraction, the different types of muscle contraction and the terms that we use for these types, the three different types of skeletal muscle fibres, and then the three different energy systems which actually fuel our muscle contraction. So our first learning objective is only a short one. I want you to be able to define a motor unit. So a motor unit consists of a single motor neuron and all of the muscle fibres in which it innervates. Remembering we have a whole muscle, which is made up of fascicles, which are made up of our individual muscle fibres. So a motor neuron will extend out from the spinal cord and it will branch out to form neuromuscular junctions with one or more individual muscle fibres. Muscles that control precise movements, so really skilled, delicate movements, will consist of small motor units. So where one motor neuron will only innervate a few muscle fibres. So think about the muscles in your voice box, in your eye, in your fingers, where you need really precise control of those movements. In muscles which are responsible for large scale or really powerful movements, but where coordination is less important, these muscles will consist of fewer but larger motor units. So for example, the muscles in our legs will have one motor neuron, which may innervate up to 2,000 or 3,000 different muscle fibres. So a motor unit is a single motor neuron and then all of the muscle fibres that it innervates. So all these red ones here and this red motor neuron here is one motor unit. Learning objective number 2 is to then begin to describe the process of muscle contraction, starting with the events that occur at the neuromuscular junction. So when we think about muscle contraction, it all happens really quickly and it appears a very straightforward process. However, a lot does need to happen or to occur for a muscle contraction to actually take place. And when looking at all those processes that need to occur, I like to break it down into 3 steps. And so those are these 3 steps here. So the events that occur at the neuromuscular junction, excitation contraction coupling, and then cross-bridge cycling. So step 1 is the events that occur at the neuromuscular junction. Remember, the neuromuscular junction is the junction or the synapse or meeting between a motor neuron and a muscle. Here, an action potential will travel along that motor neuron. It then needs to cross the synapse to innervate the muscle. And so we have looked at these events that occur at a neuromuscular junction really briefly in the nervous system lectures, but we'll look at them again in more detail and I guess with more context today. The second step of a muscle contraction is excitation contraction coupling. And so this occurs when the action potential has crossed the synapse, it's reached the plasma membrane of the muscle or the sarcomere of the muscle, and it causes release of calcium within that muscle. Step 3 is then cross-bridge cycling. And so this happens when that calcium has been released into the muscle during excitation contraction coupling. The tropomyosin moves away from the binding sites on actin. Actin and myosin can bind, they form cross bridges, and the muscle can then shorten or contract. And so we'll go over each of these 3 steps in a bit more detail. So this learning objective is focusing specifically on that first step, so the events that occur at a neuromuscular junction. And in this image here, we have a motor neuron We have the synapse or the synaptic cleft, which is that little bit here, the space between the neuron and the muscle, and then we have our muscle fibre. So all of these combined being our neuromuscular junction. So when we want a muscle to contract, an action potential will travel down along the axon of a motor neuron to the axon terminal, so to the very end of that motor neuron. The change in membrane potential in that axon terminal will cause voltage-gated calcium channels to open. This will allow calcium to enter that neuron or the axon terminal, going down its concentration gradient. Calcium in the axon terminal will then signal these synaptic vesicles to move down towards that synaptic cleft. Remember, our synaptic vesicles are full of neurotransmitter, so a chemical messenger. That neurotransmitter moves towards a membrane. It will bind to this membrane and then release its contents, so release the neurotransmitter via exocytosis. In the case of a muscle, this neurotransmitter is always acetylcholine. You may see that written as ACH. The neurotransmitter will then diffuse across that synaptic cleft and bind to our ligand-gated sodium channels on the sarcolemma of the muscle. And then we have our second step here. So we will have our excitation-contraction coupling. So going through that process again, we have an action potential coming down a motor neuron. That change in membrane potential causes our voltage-gated calcium channels to open. Calcium moves into the axon terminal down its concentration gradient. The calcium in our axon terminal causes our synaptic vesicles full of acetylcholine to move towards the membrane. They will bind with the membrane and spit out that acetylcholine via exocytosis. Acetylcholine diffuses across that synaptic cleft and binds to our ligand-gated receptors or our ligand-gated sodium channels on the sarcolemma of the muscle, the plasma membrane of our muscle. So if I ask you to describe the events that occur at a neuromuscular junction, these are the events that I would like you to talk about. So now the second step in muscle contraction is that excitation-contraction coupling, and the term excitation-contraction coupling simply describes the rapid communication between electrical events that occur in the plasma membrane of a skeletal muscle fibre and causes the release of calcium from the sarcoplasmic reticulum, which then leads to muscle contraction. So it's taking the electrical signal from the action potential and causing that release of calcium within the muscle. So in this image, we have our axon terminal here. This is carrying a nerve impulse or an action potential. We have a synaptic cleft, which is that little junction here. And then we have a muscle fibre, which is our pink box, surrounded by our sarcolemma. So the plasma membrane of the muscle, which is the blue outline. So first, in excitation-contraction coupling, we have the nerve impulse or the action potential again moved down that motor neuron, triggered the release of the neurotransmitter, and as I said, this neurotransmitter is always acetylcholine. So that's what's happening here. That acetylcholine will bind to those ligand-gated sodium channels on the sarcolemma. That opens up these ion channels, we have an influx of sodium, and then we have that action potential moving through the muscle. Now, the action potential moves along the sarcolemma and then down the T-tubules. So remember, the T-tubules are those invaginations or those little dips or indents of the sarcolemma that surrounds a muscle fibre or surrounds a myofibril. This causes the release of calcium from the sarcoplasmic reticulum and the terminal cisternae into the sarcoplasm or into the cytoplasm of the muscle. Calcium then binds to troponin on our thin filament. That causes the tropomyosin to move out of the way, and it exposes the binding site for myosin on the actin. Our myosin heads can now bind to the actin. They pull the thin filaments toward each other, towards the centre of the sarcomere, and this is what is considered muscle contraction. So the muscle is shortening and tension is being produced. The action potential then stops and calcium is actively transported out of the sarcoplasm, so against its concentration gradient. Because there is no longer calcium bound to the troponin, the tropomyosin will move back into place, covering the myosin binding sites on actin, and this causes our muscle to relax. So a lot going on in here. We'll do another recap. So we have action potential coming down our motor neuron, triggering the release of the acetylcholine, diffuses across the synaptic cleft and binds to our ligand-gated receptors on the sarcolemma. So this is what's happening at the neuromuscular junction. The binding of that acetylcholine to the receptors on the sarcolemma causes an influx for sodium to come into the muscle. That causes an action potential to run along the sarcolemma and down the T tubules. The action potential causes the release of calcium from the sarcoplasmic reticulum and the terminal cisternae into the sarcoplasm. Calcium can then bind to troponin. Troponin moves the tropomyosin out of the way. So now our actin and our myosin can bind. Our myosin will pull the actin towards the centre of the sarcomere, so shortening the muscle or allowing muscle contraction to occur. The action potential stops and we have calcium transported out of the sarcoplasm. With no calcium present, that tropomyosin moves back into place, blocking those binding sites on the actin. Because we have no cross bridges formed, or because the myosin and the actin can no longer bind, our muscle relaxes. So these are the events that occur in excitation-contraction coupling. The really important part is that the action potential triggers the release of calcium. The calcium binds to the troponin, which moves tropomyosin out of the way. Our myosin and our actin can bind, and our contraction can occur. So then the last step in our muscle contraction is cross-bridge cycling, and we did talk a little bit about cross-bridge cycling in that learning objective there. But a cross-bridge is formed when myosin and actin bind. So a cross-bridge is just when myosin binds to the actin. But I want to go into a little bit more depth about cross-bridge cycling in this learning objective. I want to show you exactly how this works and the energy requirements for this process. So this is a figure describing the process of cross-bridge cycling, but now instead of focusing just on the release of calcium and the troponin moving the tropomyosin out of the way, we're going to just look at the forming of the cross-bridges themselves. So if we kind of start here, not at the top, we kind of start here and it's a circle. So this process is going to repeat and repeat, so it doesn't really matter where you start. The first step in quotation marks is that our myosin heads, so this is our myosin here, this is our actin here. Our myosin heads hydrolyze ATP and they become reorientated and energized. So what that means is that our myosin heads contain an enzyme that breaks down ATP. The breakdown of ATP causes the myosin heads to move from this position into this position here. In this position, they're now ready to bind to the actin, and that's why we call this position reorientated and energized, because they're ready to bind to that actin. So we have our myosin heads. They contain ATPase, or an enzyme that breaks down ATP. When that ATP is broken down, it moves the myosin head back into this position so it can bind with our actin. From this position, as I said, the myosin heads can then bind to actin and it forms the cross bridge. When that cross bridge is actually formed, so when that myosin head binds to the actin, we release a phosphate. So that's what's occurring here. The cross bridge is actually being formed and we've released a phosphate molecule in that process. From there, our myosin heads will then rotate, so they go from this position, they kind of clock forward to pull the thin filament towards the centre of the sarcomere. This is what we term the power stroke. This is what actually shortens the muscle and creates that force or that tension. And this power stroke is fueled by the release of ADP from that myosin head. To then cause the release of these cross bridges, so we actually need to release the myosin from the actin so that we can have relaxation of the muscle, or so this process can happen again, we actually need a new ATP molecule to bind to that myosin head. So this little part here does seem to trick people. You actually need ATP to cause muscle relaxation, not necessarily muscle contraction. So if you think about it, this is why when people die and ATP is no longer being produced, the body will become stiff and go into that state of rigor mortis because there's no new ATP being produced to attach to the myosin head so this cross bridge can be released, so that muscle contraction can stop. So for our cross bridge to be released, we need to have a new ATP molecule bind to that myosin head so that we're no longer having our myosin bound to our actin. So looking at this process again, we have an enzyme on our myosin head which breaks down ATP. That breakdown of ATP will reorientate and re-energize our myosin head. So now it's ready to bind to our actin. Our myosin and our actin will then bind. We have our release of the phosphate. We then have our power stroke occurring, so that's when the myosin head goes from this position to this position. It moves the thin filament closer towards the centre of the sarcomere, and it's powered by the release of the ADP. So we have ADP when we break down ATP. To then release the muscle for this process to happen again, or for our muscle to actually relax, we need a new ATP molecule to bind to our myosin head. And so, as I said, this process is called cross-bridge cycling. So this entire process will continue to occur for as long as the muscle cell is being depolarized. So as long as we're getting that stimulus from the action potential and that calcium is being released into the sarcoplasm. Okay, so moving on from how muscle contracts to looking at factors that increase the force of muscle contraction, or how strong a muscle contraction we can produce. So there are 4 factors that we're going to focus on today that all increase the force of muscle contraction, but essentially all of them boil down to how many cross bridges can be formed. So how many myosin heads can actually bind to actin. The more cross bridges that are formed, the greater the force that can be produced. So the 4 factors that we'll look at in more depth are the number of muscle fibres that are recruited or innervated, the size of these individual muscle fibres, the frequency of muscle stimulation, or how frequently we send action potentials to that muscle fibre, and then the resting length of the sarcomere, or of the muscle in general. So taking a first look at the number of muscle fibres recruited, so this should be fairly self-explanatory, but for some movements we only need to recruit a small amount of muscle fibres because we don't need to produce a large amount of force. So think if you're holding a pen to write some notes, if you're playing a piano, if you're doing your hair, for these types of movements, we only recruit small motor units in which the motor neuron innervates only a few muscle fibres because we don't need to produce much force or much tension within the muscle. The less muscle fibres that are innervated, the less muscle fibres where calcium is being released into the sarcoplasm, the less calcium that is binding to that troponin and moving that tropomyosin out of the way, and therefore there's less myosin and actin actually binding. So the less cross bridges that are being formed. And of course, this goes the other way. So if we have more muscle fibres being recruited, so we're recruiting either more motor units or larger motor units, so we have more muscle fibres receiving that action potential, we will have more cross bridges being formed and a greater amount of force or tension produced within that muscle. We then have the size of muscle fibres. So with training, individual muscle fibres can actually increase in size. That increase in size will increase the number of myofilaments. The increase in myofilaments means more cross bridges, so more myosin and actin can bind, and those more cross bridges can be formed. So this increase in muscle fibre size is called hypertrophy, where the muscle fibres themselves actually get bigger. We can also have hyperplasia, which is where the muscle fibres don't actually get bigger, but we have more of them. This doesn't tend to occur as much in skeletal muscle. So we typically just have an increase in that muscle fibre size due to an increase in those number of myofilaments. More myofilaments, more cross bridges can be formed. We then have the frequency of stimulation, or how often a muscle fibre is innervated or is receiving an action potential. So if we look at this graph here, so along the x-axis, along here, down the bottom, we have the different types of stimulation. Along the y-axis, we have the force of muscle contraction, so higher being a stronger muscle contraction. So first off, we have a single muscle twitch, and a muscle twitch is the brief contraction of all muscle fibres innervated by a single motor unit in response to a single action potential. So if we have one action potential, it'll be the very brief, quick contraction of all of the muscle fibres that motor unit innervates. So obviously, with innervation from one single action potential, we will only get a small amount of force being produced. If we then have two action potentials, one straight after each other, so one action potential, two action potentials, this level of force summates. So hopefully you remember that term summates from our nervous system lectures, but it just means joins together or adds together. And we will get a slightly stronger contraction. So this is because the muscle doesn't fully relax and not all of the calcium will actually leave the sarcoplasm after this first action potential finishes and this second one starts. So we're going to have that summation or that adding together of that force because there's more muscle fibres, more cross bridges being formed. We then have what we call unfused tetanus. So tetanus just means a sustained muscle contraction. So an unfused tetanus, also sometimes called incomplete tetanus, is when muscle fibres do not completely relax before the next action potential. However, there is partial relaxation of muscle fibres between the twitches. So between these action potentials, we will have some relaxation, but not full relaxation of that muscle. And so again, this force will summate, and for each subsequent action potential, you will have a higher and higher level of force. We then have fused tetanus, also sometimes termed complete tetanus. So this is when there is no relaxation of the muscle between the subsequent action potentials. So the action potentials will occur more closely together. So this is a scenario where we're going to reach maximal force production because no relaxation is occurring between those twitches. We're just getting more and more and more muscle fibres being recruited until we reach our maximum. And until that either action potential stops or we have no calcium in the muscle to fuel that excitation-contraction coupling and that cross-bridge cycling. And then lastly, we have the length or the stretch of the sarcomere or the muscle. And this is often termed the length-tension relationship. So this refers to the concept in which there is an optimal level of stretch of a sarcomere where the maximum number of cross bridges can be formed to produce the most amount of force. So if we take a look at this graph, so down here we're looking at the sarcomere length, so how stretched the muscle is. This part here, if we think about these red bits as being our myosin and the blue bits as being our actin or our thick and then our thin filaments. So this is showing that the muscle is already partially contracted. Here we're looking at the muscle when it is at rest. So we've got that overlap of the myosin and the actin filaments. And then this is looking at the muscle when the muscle is stretched. So we have less overlap of that myosin and the actin. Along the side, along our y-axis, we have the force or the tension being produced as a percentage of our maximum. So when our muscle is partially contracted, so our myosin has pulled, or our thick filaments has pulled our thin filaments towards the center of the sarcomere, we're not going to have the maximum number of cross bridges being formed because we've got overlap of these thin filaments. So because we've got that overlap, these actin and myosin that are in this region here can't form cross bridges. So we're reducing the number of cross bridges, so therefore our force that we can produce is going to be less or not maximal. In this middle portion here, this is what we call our optimal length-tension relationship. It's when the muscle is either completely relaxed or actually slightly stretched, so up to about 120% of its normal resting length. And that's where we have the maximal overlap of our thin and our thick filaments. So the greatest number of those myosin heads can actually bind to the actin filament and create the maximum number of cross bridges. Going then the other way, we have our muscle which is beyond that 120% of resting length, so we've stretched it even further. And when we stretch our muscle, the myosin and the actin go further away. We have less overlap, so less myosin heads can bind to the actin filament, less cross bridges being formed, and less tension or force can be produced. Okay, so learning objective number 6 is then looking at the various types of muscle contraction and the different terms that we use for these various muscle contractions. So the next few slides is going to have a strong focus on some terminology. So to begin with, we aren't actually looking at those particular types of muscle contractions, but the muscle that is doing the most amount of work in a muscle contraction. So for this muscle, we use the term agonist. The agonist is the muscle that is contracting or shortening during a muscle contraction, and it's producing the most amount of force. So for example, if we think about a bicep curl, so you're curling up your elbow, you're flexing your elbow, the agonist muscle in a bicep curl would be your biceps brachii. It's shortening, it's producing the most amount of force. The opposite of our agonist is then our antagonist. In the context of a muscle contraction, the antagonist muscle is a muscle that is relaxing or it's lengthening during a contraction to allow a movement to occur. So if we think about a bicep curl again, if you were doing a bicep curl and your triceps brachii was your antagonist muscle, if this muscle didn't relax or it didn't lengthen, the agonist muscle or the biceps brachii wouldn't actually be able to perform the movement. So while one muscle is contracting, we typically need another muscle to be lengthening or relaxing to allow that movement to occur. So this is our antagonist muscle. We then have our synergist. So our synergist muscle is the muscle that assists the agonist muscle. So it moves the joint in the same direction. It is a contracting or a shortening muscle, but it's not the prime mover. So it's not doing the bulk of the work or producing the most amount of force. Again, let's think about our bicep curl. This might be the brachialis muscle. So it's not producing the most amount of force to produce that action. It's just kind of helping out. We then have the term muscle tone. So muscle tone refers to the continuous and partial contraction of a muscle or a group of muscles. So when we were talking about the functions of the muscular system in the previous lecture, we talked about how some of our trunk muscles, for example, will stabilize the body and hold us in a steady upright position. And this is because these muscles have muscle tone, so they're continually slightly contracted to hold us in that upright position. Now, there are certain medical conditions where someone will have low muscle tone, and so an individual in that situation will seem quite floppy and less stiff than a person with normal muscle tone. Then finally, we get to our different types of muscle contractions, and the first one that we'll look at is our isotonic muscle contraction. So this type of muscle contraction occurs when a muscle produces force either while a muscle is shortening, so this is what we call a concentric isotonic muscle contraction, or while it is lengthening, which we call an eccentric isotonic muscle contraction. So an example of this concentric isotonic muscle contraction, again, would be that bicep curl. So the biceps brachii is producing the force while you're lifting a weight, and that muscle is shortening. So this is the most common type of muscle contraction. An example of an eccentric muscle contraction is, say you are again at the gym, you're doing the chin up, you've pulled yourself up, and then you're slowly lowering yourself back down. So you're controlling that movement, you're producing force or tension in the muscle as you move back down so that you don't just fall to the ground. So this muscle is lengthening while you're doing this. Now, this type of muscle contraction, our eccentric isotonic muscle contraction, is said to produce more damage to the muscle, because instead of the actin and the myosin heads binding and being pulled towards the center of the sarcomere, they're going in the opposite direction. So they're actually being pulled apart while they're trying to hold on to each other to be able to maintain that muscle force and resist the load. So because of this additional muscle damage that occurs, it's also believed that this type of contraction will produce greater training adaptation, which not the focus of today, but just kind of a bit of an interesting little fact. So our isotonic muscle contraction is when we have contraction and force or tension being produced either while the muscle is shortening, which is a concentric contraction, or lengthening, which is our eccentric contraction. And then lastly, we have an isometric muscle contraction. So this is where a muscle is producing force, but the muscle isn't changing length. So think about doing a wall squat or a wall sit where your leg muscles are producing force to hold your body up, but because there's no movement, the muscle stays the same length. Same with, say, a plank or any kind of exercise really where you're holding a static position. You still need to produce tension in that muscle to hold yourself in that position, but the muscle isn't changing length. So just to recap, I guess, those three different types of muscle contractions, and again, we're going to look at a bicep curl. So we have our concentric isotonic muscle contraction, where a muscle shortens while it produces force. So our bicep brachii is undergoing a concentric isotonic muscle contraction when we're lifting that weight up. It's shortening. We then have our eccentric isotonic muscle contraction, where a muscle lengthens while it produces force. So in this example, we're now slowly putting that weight back down. So our biceps brachii is lengthening while producing the tension or the force. We're not just letting that muscle, that weight drop. And then we have an isometric muscle contraction, where we're producing force or tension in the muscle, but that muscle isn't changing length. So in this case, we're just holding that weight and holding our elbow at that 90-degree angle and not moving. We still need to produce force to hold that weight in this position, but our muscle isn't changing length. Now, the last two learning objectives are changing track a little bit, and we're going to first look at the 3 different types of skeletal muscle fibres, and then we're going to look at the 3 different energy systems that actually fuel our muscle contraction. So starting with the skeletal muscle fibre types, and there are 3 that we're going to look at. Research is always discovering new things, and there's actually believed to be more muscle fibres than this right now, but these are, I guess, considered the 3 primary and the most accepted ones, the ones that you'll read about in your textbook. And so we're going to look at these 3 different types of skeletal muscle fibres by comparing their characteristics in a table. So to begin with, the 3 different types of skeletal muscle fibres that we'll look at today are our type 1 fibres, our type 2 fibres, and our type 2X fibres. So using our Roman numerals here. So when we describe these 3 different skeletal muscle fibre types, or we classify a skeletal muscle fibre, it is done so based on 2 characteristics. How fast do these muscle fibres contract relative to the others? And how do the muscle fibres regenerate or produce ATP to actually fuel that muscle contraction? And so that's what we have in this description row here. So our type 1 fibres are considered slow oxidative fibres. So these fibres contract relatively slowly, and they use aerobic respiration, which we're going to talk about in the next learning objective, but they require oxygen to contract. Our type 2A fibres contract quickly, and they primarily use aerobic respiration, but they can also switch to anaerobic respiration, also called glycolysis. So again, we're going to talk about that process in a second, but they contract quite quickly, and they can use oxygen, but they can also go without. Our type 2X fibres also contract quite quickly, but they primarily use anaerobic glycolysis as their muscle energy source. This second row along here is then describing how much force or tension each of these skeletal muscle fibres can produce. So our type 1 fibres produce quite a low amount of force. They're slow to contract, and they produce quite low levels of tension. Our type 2A produce a moderate amount of force or tension, and our type 2X produce the highest amount of force or tension. Comparing their ability to resist fatigue, our type 1 fibres have high fatigue resistance. So this means they can contract all day, producing their lower levels of force without getting tired. So this is important for muscles that we use all of the time. So again, think back to those stabilization muscles in the trunk, or say the muscles in our hands or our feet and calves that we kind of use, day in, day out, walking around or doing day-to-day activities. Our type 2A fibres have similar levels of fatigue resistance, so quite high levels of fatigue resistance, while our type 2X fibres are have quite low levels of resistance to fatigue. So these fibres can produce a high amount of force, but they can't repeat that activity very often. Our time to peak force simply refers to the time between an action potential being set and the peak force or tension being produced in that muscle fibre during a single muscle contraction. So type 1 fibres are quite slow from that electrical stimulus to that peak muscle contraction. So roughly 80 milliseconds, which is quite fast, but relatively it's quite slow. And then our type 2A and our type 2X are quite fast, so roughly 24 milliseconds. And then lastly, the duration of the contraction, so the duration between a muscle fibre being innervated to reaching that peak tension to then completely relaxing. So for our type 1 fibres, this is quite slow. So again, it's slow to reach that peak force, slow to relax. It produces quite that low level of muscle force, but it can contract all day without getting tired or without being fatigued. Our type 2A and our type 2X fibres are quite fast to reach that peak force. They're also fast to relax, and then they produce that moderate and that high level of tension within the muscle. And then our last learning objective is to describe the 3 energy systems which fuel our muscle contraction. So to begin with, our muscles use ATP, or adenosine triphosphate, to fuel muscle contraction. So we spoke about this when we looked at our cross-bridge cycling. When we break down ATP, so particularly as part of that cross-bridge cycling, but also elsewhere within the body, ATP breaks down into ADP, or adenosine diphosphate, so 2 rather than 3, and a phosphate ion. When we break down ATP, energy is released, and this is the energy that is used for muscle contraction. ATP is the only source of energy that we can use for this process. So there are 3 energy systems that produce ATP for us to use for muscle contraction, and the first one is called the ATP-CP, or the adenosine triphosphate creatine phosphate system. So creatine phosphate, you might also see written as PC or phosphocreatine, exactly the same thing. So a creatine phosphate is a substance that is stored within the muscle, and it helps regenerate ATP or turn our ADP back into ATP. So when creatine phosphate is broken down, it becomes creatine and a phosphate. That phosphate then combines with our ADP, and it forms ATP and creatine. Now, because this creatine phosphate is stored within the muscle, no oxygen is required for this process to occur, so it all occurs very quickly. So when we need a quick burst of energy, this energy system is what's going to produce that ATP. However, the total store of creatine phosphate within the muscle is very small. It only produces 1 molecule of ATP per cycle, and so it can only provide energy for approximately 15 seconds of muscular activity. So this system will fuel muscular contraction in activities that are really high intensity but are of short duration. So think things like sprinting or, say, weightlifting. Our second energy system that we have to fuel muscle contraction is anaerobic glycolysis. So some of these terms you will remember from our different skeletal muscle fibre types. So in this system, glucose is broken down, which is called glycolysis, to form 2 ATP molecules and 2 pyruvic acid molecules. Those pyruvic acid molecules are which then converted into lactic acid. So you might have heard about lactic acid being a byproduct of muscular contraction. Now, the process of anaerobic glycolysis is quite complicated. It involves the actions of a number of different enzymes, and it's well beyond what we expect you to know in this unit. But what I do want you to know is that this process happens relatively quickly. Therefore, it can be undertaken without the requirement of oxygen, hence anaerobic, doesn't need oxygen. It provides energy for muscular contraction for activities lasting between 15 seconds and roughly 2 minutes. And so think about your longer running events, say a 400 metres or things like repeated efforts in a team sport. The last energy system that our body uses to produce ATP for muscle contraction is the aerobic system. The aerobic system is used to produce ATP at rest and during low to moderate intensity exercise. So about 95% of all of the ATP we produce for muscle contraction actually comes from this energy system. So similar to our anaerobic glycolysis, the aerobic system is quite complicated, and it uses quite a variety of processes to actually regenerate or produce this ATP. Processes which we will not test you on. However, just in case you're doing some reading and you come across these processes are aerobic glycolysis, the Krebs cycle, and electron transport chain. And approximately 36 to 38 ATP are produced in one cycle of this aerobic system. The aerobic system produces ATP from a variety of fuel stores, including carbohydrates, fats, and protein. However, our carbohydrate and then our fats are used preferably. So if we can break down our carbohydrate or fat to produce ATP, we will do that. If we run out of those energy sources, we can then break down protein. This system does require oxygen, hence aerobic, and hence why it takes a little bit longer for this system to work. But we can continue to produce ATP basically until we have no oxygen or until our fuel stores are depleted. So we'll use this energy system while we're at rest, so we're kind of sitting at our desk doing work, or we're making dinner, our kind of day-to-day activities, as well as other lower intensity exercises, so things like going for a walk, but also things like a marathon that will go for a prolonged period of time. So again, on our last slide with the tables, because I think they're a really nice way to summarise a few slides of information into one image. So in this table, we have our 3 energy systems, and we have the characteristics of each of those energy systems that's important for you to know. So firstly, for each system, I would like you to know the fuel that system uses to produce ATP. I'd like you to tell me about the duration of energy that's provided by that energy system, the intensity of the exercise or activity for which each system can provide that energy, whether or not oxygen is required, and approximately how many molecules of ATP is produced. Now that is it for our muscle contraction and our energy. We have one more lecture to finish off the muscular system. So in the next final lecture for muscles, we are going to focus on smooth muscle and particularly how it compares to our skeletal muscle.